Isn't that faster?
If you come here.
I can go over these questions. I'll go over these questions and give you all the answers to these first ones,
that's
okay?
good.
And then we'll do the second one on here. All right. So stain, and you guys can all shout them out if you know them, so I don't have to tell you. This staining method stains the background, not the organism. Negative staining, very good. The advantage of this method of obtaining pure culture is that it takes less time and materials. Streak plates, good. An environment where the solute concentration is lower on the outside. Hypotonic, very good. Hypo means less, hyper means more. The term for an organism whose optimal temperature is between 20 and 50 degrees Celsius. Everybody got that?
Yep.
Mesophiles, good. Which reagent was used to test for the presence of starch? Okay, you should all have it. This is an example of a selective medium. McConkey, yeah. What does it mean to be selective?
Only gram-negative.
Only certain things grow on. Good. If your organism formed endospores, what color would they be?
Green.
Green, good. We talked about that. Phenol red acts as what?
pH indicator.
A pH indicator. That's why it changes color. If it changes to yellow, it's because it's producing the acid. Okay? It's a pH indicator. You can also use red cabbage. All right. Term for an organism that can tolerate oxygen but does not use it.
Aerotolerant.
Aerotolerant. Everybody's studying Robert. Okay. This medium was used to determine if your organism was motile. A sim medium, good. This is a culture medium in which the exact chemical composition is not known.
Complex.
Complex medium, good. Cyanobacteria share this trait with algae. Cell wall's one. Photosynthesis, very good. Cell wall or photosynthesis, because cyanobacteria are photosynthetic. Okay? This is the method we use to test antibiotic sensitivity.
Kirby Bauer.
Kirby Bauer, that's them. Yep. All right. This is the term for an organism that cannot tolerate oxygen.
Obligate anaerobe.
Obligate anaerobe, very good. This is the part of the microscope focuses and directs light from the lamp to the stage. Condenser.
I'm confused on that.
What part?
I thought that the iris was like the controller and the condenser was just the plate that it goes from.
Just anything underneath that stage we're going to call the condenser. Okay?
Oh, so that is the condenser.
Yeah, the whole thing underneath, yeah, and when you're adjusting the light, we're just anything that adjusts the light underneath the stage we'll call the condenser.
Okay.
Okay? All right. This is the term for an organism that requires a high salt concentration to grow. Ooh, a what?
Halophile.
Obligate halophile. Requires. Anytime you see that word require, stick in obligate. It has to have it. Obligate halophiles. All right. An agent that kills or controls the growth of microbes in general is
Antimicrobial.
antimicrobic. Antimicrobial. Yep. That's kind of a generic question. If you're viewing under the microscope, which knob do you use under high power? Oil immersion.
Fine.
Fine focus, good. This is the term for an organism that can tolerate a moderate amount of salt, but does not require it. Halo tolerant, there you go. So just remember,
So
look
if it said
for,
required, it would
look
be
for
obligate.
required means obligate. If it says tolerate, it's tolerant, good. We've also got those facultative ones, those ones that can go both ways, right? This is an example of a complex medium.
Nutrient.
Nutrient agar, yes. Because we don't really know what the nutrient is in there. It's just something that just, complex medium just grows stuff on. That's what it's for. Which reagent was used to test for the presence of catalase?
Hydrogen peroxide.
Hydrogen peroxide, very good. Give me an example of a basic stain. Crystal violet. I think that's the only one that was on the card. Did anybody have anything else they thought was a basic stain?
Saffron.
Was saffron on there?
No, and Mexican green are.
No, I meant is on one somebody's card. I think, yeah, the one you were looking for in your card was crystal violet. But yeah, all those are basic stains. Okay? Yeah. Crystal violet. Yeah. All right. If an organism can hydrolyze starch, what enzyme does it produce? Yeah, it's amylase. That one is something we didn't really get into. That's an enzyme that breaks down starch molecules into smaller sugars. It's what's in your saliva. All right, this type of medium allows certain organisms to grow, but inhibits the growth of others.
Selective.
Selective, right. Organism that grows in hot stuff.
Thermophile.
Thermophilus. What is the organism that is a thermophile?
G. Sterothermophilus.
G. I like how you said G. Yeah. G. Sterothermophilus. Yes. It says thermophile in its name. Yes.
We have to write that, if that's a fill in the blank?
Yes, in italics.
Then spell it. I've never heard that. Spell that one.
Don't worry
It's
about
on my card.
We can look at that
it.
sometime.
I mean, yeah. All right.
If we actually spell that wrong,
It's only like half credit off, like 25 points.
I would never get it.
Don't worry about it. Just know what that one is. All right. An agent that kills or controls the growth of microbes and is safe for use on human tissue.
Antiseptic.
Antiseptics, good. What are the three lenses called?
Objective.
Objectives, very good. What's an organism that requires oxygen?
Obligate. Arrow.
Obligate arrows, good. What's a colonial green algae?
Who knows?
Did anybody have that one?
Cyanobacteria.
No.
Photosynthesis.
Colonial, starts with a V.
Volvox.
Volvox, yes.
How do you spell it?
right then.
V-O-L-V-O-X.
I was wondering what that one was on my,
Volvox.
on my
What?
bingo card.
Yeah.
I have it. It's called.
It's that round one that had the
Green
smaller
algae.
round ones in it. It's the big round one that had smaller round colonies in it. All right, an environment where the solute concentration is higher on the outside than the inside.
Hyper.
Hypertonic. What does it mean when all the lenses will stay focused?
Focal.
That's parfocal. That's right, parfocal. And lastly, if your organism's gram-positive, what color is it?
Purple.
Purple. Gram-positives are purple. All right. So clear your boards, and we'll do the second stack. Sorry.